And I also like to thank Carla and the organizers for inviting me, and for Juliana for suggesting me. I know we heard a little bit about imposter syndrome earlier today, and it certainly is a bit intimidating to get up here and talk to everybody after all these great talks. And we're going to talk a little bit about something completely different. And this talk sort of spun out. I don't know if people can see the T-shirt that I'm wearing. This is the famous photograph from Rosalind Franklin. And so this talk spun out of a talk that I gave at the Australian Genomic Technologies meeting a couple of years ago. So obviously I was preaching to the choir there. But that was right after an American Society of Human Genetics meeting, where as part of the meeting they gave out buttons of important moments in science, particularly in genetics and genomics. And one of those buttons was this picture. And the number of graduate students who did not recognize this picture was horrifying. So it was definitely something that I thought was useful, but I think it's also, for me, also personally engaging, because I've spent most of my career trying to develop technology and tools for the biological community to use, and these sorts of things don't often get a lot of love, right? Like we've seen a lot of talks this meeting talking about the impact of cell phone technology. But I would actually argue cell phone is an application. The technology that enabled cell phones are things like lithium batteries and integrated circuits, which are a little bit boring. And so what I want to do is talk a little bit about some of the technology that leads to the applications that we care about. Just to make sure everyone has my disclosures. And so as mentioned, I'm at a company called Inscripta. And this is a quote that we use a lot from Sidney Brenner about the progress in science depending on new techniques, new discoveries, and ideas in that order. And so I think it's actually very interesting that while Sidney was a great tool builder, he did not actually receive his Nobel Prize for tool development. He received it for the biological discoveries, because that's what really excites people. And we won't, he shared this Nobel Prize with two other gentlemen who were instrumental in this work, but I would argue that while the biological discoveries were really interesting, I think one of Sidney's greatest contributions, and he certainly had many, was the development of model organism and the genetic toolbox around the roundworm C. elegans. And so there's a picture of what this little roundworm looks like. And it's a lot of work to actually take a wild organism and turn it into a model organism, to put the genetic switches and the tools that you need to analyze that. And the work that he did here with his colleagues has enabled lots of researchers after him to make new biological discoveries. And in fact, we now even see companies being developed to use C. elegans to understand variants that are being identified in the genomes of children in the NICU unit at the Rady Kingsmore Hospital in order to try and better interpret their genome. So I would argue that even this sort of unsexy work of doing technology development is critical for the advancement of science, because we can only see as good as our tools allow us to see. And so when I started this talk a couple years ago, I did something that I often will do, is I'll reach out to Twitter for some help and advice to kind of see what other people in the field think about technology and the things that have moved their field. And I certainly got a lot of interesting comments from people, and they were really kind of all over the place, from basic molecular biology techniques to high throughput computing. But a couple of these really got me thinking. And so one of these was about microscopy, right? And there were a couple of instances here. I certainly agree the microscope was instrumental in starting microbiology and thus all of microbiome. But there are two things that I sort of also took about this. I would actually argue that microscopy was also instrumental in genome research. And we'll talk a little bit about that. And also, I was surprised to learn, because we all learned in school about Leeuwenhoek developing the microscope, but there was actually a ton of work that went on before he actually made the changes that really made modern microscopy actually tenable, but that work was necessary for him to make those advances. And so what I'm going to do during this talk is actually talk about some ideas that existed in science, and the development of new technology allowed us to change our ideas about this, right? And so we did learn a little bit earlier from Rachel about how DNA is boring compared to RNA and proteins. Totally get this. But this is one of the things that really led to this notion that the genome can't possibly be complex enough to be the unit of heredity. And so when I talk about microscopy leading into genomics, this is what I mean. So one of the technologies that people are really excited about today is single-cell genomics. This is the original single-cell genomics. This is being able to look at the genome. So these are all the chromosomes, and these are human chromosomes that are in the cell of an individual. Now we'll notice a couple of things here. There are two copies of each chromosome, although in this case the copy is a little bit different because this is from a male, so this is an X and a Y chromosome. So we knew about chromosomes for a while, like they predated Mendel. An interesting fact I also discovered was that even prior to there really being buy-in about the importance of the genome, there had been some work done both in terms of understanding that in bacteria, DNA could be the transformative factor, but also Nettie Stevens had done some work in insects and had discovered that the sex chromosome, there are two chromosomes that could determine the sex of these insects. Now, she often does not get credit for this work. This is often credited to Edmund Wilson, but she was the one who did a lot of the seminal work here. But even as early as the early 1900s, we didn't actually know how many chromosomes a human had. This seems like this is something that we all think we know about now, but I think part of this stems from the fact that people just didn't think chromosomes or DNA were all that important. And I would argue this photo changed that, right? This photo by Raymond Gosling, who was with Rosalind Franklin, which was using a technology. It wasn't even a super new technology, but it was a technology applied to a new problem. And that was, could we use this to understand the structure of DNA? And so as soon as Watson, Crick, and Williams got a hold of this data, it became clear what the structure of the DNA is, and that really excited everybody because we could now understand, oh, this structure means that we can replicate DNA faithfully. And so even though the genetic concepts of genes had been known, now we had insight into the mechanisms by which these could be transcribed and translated and maintained. And so this really, I would even argue, set off additional work. So not long after this, we actually did have better techniques to allow us to understand how many chromosomes a human individual has. And this sort of staining technique, I was actually surprised to see, still is used quite a bit today in the genome editing field because this is still considered the gold standard for understanding that you have a structurally intact genome, even though this is a very low throughput technology. But it was not long after this, in fact, that additional discovery was made, and that is that having an additional copy of chromosome 21 led to a human phenotype called Down syndrome. Again, this is another example as well where an individual is typically given credit for this, but this actually was primarily the work of someone named Dr. Marc Gauthier, who was a scientist in France, and she had been working hard on trying to develop and regularize these techniques in her laboratory so she could look at patient samples to try and make this association. But she did not have the resources or the quality of microscope to actually take pictures to get good data, and so she had to share that data with Dr. Lejeune, who then went off and told the world about this. And people who are more interested in this can go to Rosie Redfield's blog and learn a little bit more about this story as well. So even with relatively simple technology, we've been able to change this notion that the genome can't possibly be complex enough to be the unit of heredity, to not only is the genome the unit of heredity, but observed changes in this can be correlated with phenotypes, right? This is the core of what human genetics is about, is can we correlate genotype or changes with DNA in phenotype? And so we'll move on to something a little bit more modern, although I realize now it's been going on for quite a while. And this is also pretty, I would say it's integral to my career as well. So I started graduate school when the Human Genome Project officially started, and I was working in a laboratory that was doing some of the original mapping for the Human Genome Project. And so I think it's interesting because so many people don't actually think about the human genome assembly or the reference assembly. I've worked on this for a large majority of my life, and it is the most boring thing. When I tell other scientists I work on doing the human genome, their eyes start to glaze over. Because it's a tool, right? Like it's a tool that people use to facilitate their research, and they use it every day, but they don't think about it. It's like getting in your car. But there are certain things, decisions that were made early in the human genome that impact everyone who's using the reference assembly today. And this notion was that despite the diversity of humans that we see in this population, the thought was we really only need to assemble one human genome, and in fact, we only need a haploid representation. So remember I showed you the picture earlier where we have 2 copies of every chromosome? The reference assembly only represents 1 copy of each sequence. And this will be fine for analyzing everybody. Now, it's easy to be critical in retrospect, but I think it's important to understand why this idea was thought. And part of it was because of the technology that was available. So it's important to keep in mind that the Human Genome Project was not just about making a reference assembly. It was a technology development project. And so, we hear all these talks about how it cost us $3 billion to do this first assembly and now it's so cheap. Well, that's because we had to develop the technology to do this during the project. In fact, when the Human Genome Project started, we didn't even know how we were going to finish it because the sequencing technology was pretty low. And so we were thinking about things like, we want to be able to sequence 500 megabases a year. Well, now, as I think Chris pointed out the other day, the Broad sequences 3 gigabases every 6 minutes. So this is, these numbers are not even things we even think about. But the other notable thing here is that the other goal was to map 100,000 SNPs, or single nucleotide polymorphisms. These are single base pair changes in the genome. It's not a whole lot. And this was born to the fact that there was a prevailing notion, I think partly limited by the technology that we had, was that the DNA sequence in everybody was really not that different. So most of these differences were going to be driven by these SNPs. And so the way you put the reference assembly together at its heart is you take all these reads, so you generate your sequence reads. You don't know which chromosome those reads came from in that individual, but you can pile them all together, and then you try and make this consensus at the bottom. Now, one thing that will happen is when you look at the individual reads, you will see differences. And in fact, you can see this T and this A go together all of the time in all of these reads, so they are on the same piece of DNA. But yet they might get mixed in this consensus because what this consensus is trying to do is average across all of these reads. Now, I would certainly argue, in fact, that this notion of trying to average across all of the haplotypes is absolutely fine if the only type of variation that you have to think about are these single nucleotide changes, right? The reads align nicely. This red here indicates a difference. These two individuals are the parents of this individual, so you can see they each had one allele that had the red and the other that had the gray, and their child got both. The problem is, this is not the only type of variation that we have to deal with in the human genome. And it was because, in fact, the availability of this, despite its flaws, high-quality reference assembly and the development of new technology allowed us to get better insight into that. And so one of the early technologies was this idea of array comparative genome hybridization. And so what you could do is you could spot pieces of DNA on a slide. You then take DNA from 2 different sources and you color them differently so you can track them. You hybridize them together, and so the parts of the genome that have the same copy in its genome kind of wash each other out. But if you have regions that have higher copy in one than the other, then you can get a different color, and you have this colorimetric readout effectively. And this sort of assay is very good for detecting this kind of situation. So here we have chromosomes from several individuals. We can see this is the ideal scenario where this individual has 3 copies of this sequence on both chromosomes. This is the easy case. But we see many other scenarios where even within the same individual, different chromosomes can have different copies of DNA sequence. Now, it's not that this type of variation was not known at the beginning of the Human Genome Project. It was just thought to be rare and predominantly associated with highly penetrant Mendelian disorders, typically intellectual disability disorders. But because of the availability of the human reference assembly, which I would argue was technology, as well as some of these high-throughput detection assays, we began to realize that copy number variation was actually common within the human population. Every single one of you sitting in here has millions of base pairs difference, probably between the 2 chromosomes within you, and certainly across members in the audience. This was not something that was taken into account when the Human Genome Project started. And since then, as sequencing technology has developed, we're actually learning there's lots of ways your genome can be different from one another. So it's not just copy number differences. It can be orientation differences. It can be basal sequence differences. And as our sequencing technology gets better and better and reads get longer and longer, we actually find out that there's way more of this in the population than we ever could have anticipated. And the impact of this, though, can be stark. So, I would argue we've known about this, that initial copy number variation paper was published in about 2004. And we've largely ignored tons of this for the past couple of decades because it's really hard. But here's the implications of this. And so I have a toy example here. So if you have a reference assembly that looks like this, because remember, we can only represent one haplotype, but you have a sample that looks like this. what most people think is that, oh, well, that's too bad. I'm not going to be able to analyze this guy, but I can analyze this guy and all is good. But it turns out that in many cases, and if this guy looks a little bit like this guy, the reads from here are going to align here. So not only now are you not able to analyze this one, you're now messing up the analysis of this one because, you know, an aligner, a sequence aligner is a tool like anything else, and it's a bit like a hammer. You know, it's gonna try and align things, and so if it's pretty close, it's gonna align. Now, when my group, when I was still at NCBI, was working on the reference assembly, we were actually able to do an experiment to see how prevalent this was. And so the previous version of the reference assembly, known as GRCH37, we had been working on that, so we had identified sequences that we knew were not in this assembly. We had data to support this. And so we were able to take those sequences and break them down and so they looked like short reads and then align them back to GRCH37. So we know they're not in that assembly, so none of them should have aligned. We tried 4 different alignment methodologies because we wanted to make sure it was not a methodological problem. And in every case, 70% of those reads aligned to the reference assembly. So this tells us that this is not just a theoretical problem, that this is a serious problem that is impacting genome analysis using this simple linear reference, which is what our entire tool base is built on. So I'm very excited by the fact now that there are a lot of groups, and I highlight this paper by Benedict Payton, but there are many groups that are now working on this, is we want to start a new technology project to see if we can now come up with better models and structure that let us represent population variation within a reference assembly. So we don't just have this straight line anymore, but we can now represent all of that variation. I'm even more excited that, you know, almost two decades after the conclusion of the Human Genome Project, that now NHGRI is back to funding the sequencing of human genomes and the development of this tool base to allow us to try and take the Human Genome Project now to the next level and make it a reference that is useful for the entire population, not just a small subset of the population. And so, you know, here we've gone from this notion to where we only need to sequence, you know, one person, and we have this simple linear reference to this notion that we actually really need to assemble lots and lots of genomes from lots and lots of diverse people if we want to analyze everybody. And so lastly, what I'll talk a little bit about is this idea about, and I would say there are a lot of people who still think this. I think this is an idea that I hope is somewhat in transition, which is that, you know, in our effort to understand genotype, phenotype, what we really need to do is just be able to sequence more people, right? If we sequence enough people, we're going to figure it out. But we've seen some examples. I'll walk you through. This was an example from my own past of how better tools and give us different insights into this genotype-phenotype association. So I was involved in this work to identify the underlying variants involved in the most common form of genetic dwarfism, which is achondroplasia. And so we did this in the early 90s, so this was before we even had a reference assembly, which was, there was almost a golden age of gene-phenotype association. But one of the downfalls of the way that we did work back then is if we found a candidate variant that we thought looked like it was associated with the disorder, we would look at 50 control samples. So that's 100 normal chromosomes. And if we didn't see it in those 50 people, we'd be like, yeah, we've got the smoking gun. This is good. Now, part of the reason for this is this was the state-of-the-art sequencing technology at the time. I don't know for sure that I ran these gels, but I certainly could have. So these are gels that you had to pour manually between 2 glass plates. They're very thin polyacrylamide. You then radioactively label DNA, and then you have to manually read up the gel and then type it into a computer to do your analysis. So this is not very high throughput, although I was really fast at it at the time. But I would say, you know, we were pretty lucky with achondroplasia for a couple of reasons. So we had several families where we had a de novo event, so parents were not affected, but the child was. And interestingly, achondroplasia is very unusual in this, but almost everyone who has achondroplasia has the same variant. This is almost unheard of in genetics, but it is one of the things that simplify this. But then we've had, you know, this is not the first time that we've talked about the revolution that high throughput sequencing has involved. And I like to show this because, so this is the one sequencing technology we had to finish the Human Genome Project. So here on the x-axis we have read length. So let's remember a human genome is 6 billion base pairs. when haplotype is 3. So these reads are about 1,000 base pairs each. Here we have throughput, and this is a log scale. Prior to the mid-2000s, this was the only game in town. We now have this rich ecosystem of technologies that provide us different read lengths, different throughputs, and it's really opened up what we can do with genome sequencing. And one of those things is that we can sequence more people. And so not long after some of these technologies came about, there was a project called the 1,000 Genomes Project that actually ended up sequencing about 3,000 genomes, but give or take, 1,000 genomes is a little catchier. And this was really one of the first high throughput, again, tool development projects, because many of the procedures and file formats that we use today really came out of that technology. But this group decided to take that 1,000 Genomes project and look at all of the data that had been associated just on the X chromosome with disease, most of it intellectual disability. And what they found was that 10 to 15% of the variants that had been associated with intellectual disability prior to the availability of high throughput sequencing were probably wrong associations. And that's because you could take these variants that had previously been associated, look in the 1,000 genomes data, and see plenty of people who do not have this phenotype that carry this variant. And so it really underscores the lack of technology, our inability to look at lots of people really hampered our ability to do that genotype-phenotype correlation previously. We're no longer hampered by this. There's likely over 1 million genomes that have been sequenced worldwide. So this is a plot of just the Broad Institute, so just one genome sequencing center. It's not the only one in the world. I just saw a paper that came out yesterday on bioRxiv from Decode in Iceland, where they've sequenced over 1,100 genomes using new long-read technology. So you can't even keep these slides up to date anymore. And so we can look at a lot of genomes now. And one of the things that I certainly take home from this is that sampling natural variation is actually insufficient for us to do this kind of genotype-phenotype association. One reason for this is because it's not possible for us to observe all of the variants. And so this figure, this is a really great work from Dan MacArthur's group. It's a resource called Nomad, which I would say has been revolutionary for the human genetics community, because they have aggregated genome data from lots of people, and this is also a number that's hard to keep up with right now. But they've analyzed it all in the same framework. And what we see here is these are, even despite the large number of people they've looked at, these are the percent of the variants they've observed versus what they've expected. So you just don't see all the variants you might want to see. Now, we know, because this project, even though it's starting to move into the whole genome, is still very biased towards genes or coding sequences. We know part of that is due to selection. So a lot of times genes don't like to tolerate variation because they have very important function. But this also has implications for us doing genotype-phenotype correlations. And it's because that, yes, when you look at an individual, so any of you sitting in the room, most of the variation that's in your genome is common. But if you look across the population, most of this variation is rare, because each of us will carry about 100 rare to unique variants. And so when you look at these plots that show a minor allele frequency, or this is how often do you see the rare variant, this is what dominates the population. And so what this means, and this plot, I think, from Leah Starita at the University of Washington really nicely demonstrates this, is that even when you sequence people that are highly ascertained for a genetic disorder, or they may even have a genetic disorder, you end up identifying more and more variants that you can't interpret. And this leads to this collection of what we call VUs, or variants of unknown significance, in a database called ClinVar that tracks these things. And it's because if you only see a variant in one person, that is not enough for you to say that that variant is causing that disorder in that person, even if they already have it. So there's already interest in the community to try and see if we can do functional variation. Can we create variants so that we can specifically test them in some sort of molecular assay to understand their function? And so this was landmark work from Jay Shinduri, where they took the BRCA1 gene. So we know this gene bears a large burden of breast cancer across the world. And they used CRISPR technology, gene editing technology, to create variants in this gene. And then they had a molecular assay that they could test to see if they thought if these variants are functional or not. And the challenge of this is that even though this paper was very exciting for the human community, Very few people are doing this right now because it's really, really hard to do. And in fact, in this paper, they could only do about 28% of the gene. So they only did these exons here, leaving lots of the gene that still hasn't been assayed. And that's because today, when people want to do gene editing, it's really a lot of work. We need better technology to do this. This is in its nascent phases. So most of the time, if people want to manipulate a genome, they either have to go, they can look at a lot of locations, but do pretty limited types of edits. Or they can do a lot of different types of edits, but at a small number of locations. And so if we want to be able to change this, we need to be able to make lots of edits, of different types of edits, all over the genome. And so we need to really improve the technology here. And this is why I'm really excited with my new job at Inscripta, where I've now switched from working on reading genomes and helping to make genome assemblies to now thinking about how can we create this variation. So we do have new technology that allows us to do this. So this is an instrument that people can run in their lab. They can order the reagents. It sits on their benchtop. Now, it's still early days of technology. This only works on E. coli and yeast, and I just spent the entire time telling you about the human genome. But this is how technology actually develops. And in fact, even the human genome started off by sequencing yeast, C. elegans, Drosophila, right? Like you develop technology on simpler systems. But we think the promise of this is that we can really create a lot of variation. And we can do it in both a biased way. So if we have a pathway that we're interested in, maybe there's a protein that we think is seminal in that, and we can do saturation mutagenesis on that protein. And we might even decide that we want to do saturation mutagenesis on other members of that pathway, right? We know genes don't act in isolation. We saw, nice work from Rachel earlier today working out pathways for stress. And so you'd want to look at multiple things in a genome, not just once. But the one thing that we really don't want to do is we don't want to bias ourselves to things we already know, because if there's one thing that I've learned from being in science all these years is we really don't know anything. Every time we think we know something, we find out that we're wrong about something. So we really want to be able to also think about doing unbiased assays. So can we go through and do knockouts across the genome? Can we change the regulatory sequences around genes throughout the genome? So that we can not only ask questions about things, specific questions about things we think we know, but we can also ask questions in areas where we don't really know anything. And we've actually been able to do this in yeast. So this is some data from this experiment where we are trying to improve tyrosine biosynthesis in yeast. This is a pretty well understood pathway. And what we find, though, is that we find new hits, but using both our sort of, biased assay as well as our unbiased assay. And so, this ARO4 gene has long been known to be involved in tyrosine. And so not only do we find variants that had previously been associated with increased production, we can find new variants in this well-studied protein. But we're also able to find genes that nobody knew was involved in tyrosine biosynthesis because we could do this unbiased approach right now. And so I think this notion of going from having to just observe natural variation to being able to create variation and then directly test it using whatever phenotype assay that you care about is going to be the next technology change for human genetics and genomics. And so hopefully what we'll see, and I probably should have colored this differently because I think we're still in this notion right now. We're in the middle of this transition as the technology develops and this is not that we don't need to keep sequencing. We absolutely do need to keep sequencing more humans, but we need to couple that now with creating variation that we can test so that we can make the most out of that data. So hopefully I've convinced you all that technology is really this driving engine of biology, and it is really what allows us to keep making these great discoveries. And with that, I'm happy to end. Take questions or not, depending on the time.